Today I'm going to talk to you about the brain, but really something that goes wrong in the brain. I'm going to talk about brain cancers. And I think the major point that I want you to walk away with at the end of this is that there is actually a different way to look at complicated biology problems through the use of physical science that allows you to make very discrete predictions while not worrying too much about the detailed mechanisms that are making the biology, say, work in error. And so I'm going to talk about glioblastoma multiforme today, which is it's the most deadly brain cancer in adults. It's if you get it, basically, you're going to die. There's no surgical cure. There's no effective therapy for it. And it's a prototypical heterogeneous tumor, which means that it is, people think about tumors or heterogeneous tumors being complex. This is truly complex. But a lesson I learned from my friend, who I'll introduce later, Mike Phelps, is that the word complex, in some ways that implies that you can't understand the problem. And so I don't want to use that word. A word diverse is a better word, because I actually think that one can understand this problem. So I'm a physical scientist, and I'm relatively new to working in the biological world. But something that struck me, biologists think very, very differently. And there's an analogy between biology and the Japanese postal system that I want to make. So in the Japanese postal system, there's no logical connection between an address and the actual physical location of the house. And so if you are a postal worker, the only way you can deliver your mail is to memorize your route. And so that local knowledge is very valuable, but if you are assigned a new postal route, everything you learned about your job is not useful anymore, because now you have to memorize a whole new route. And in biology, in many ways, has evolved into that world of very, very local knowledge, which is very different from the physical sciences, where you have laws that can move you from one field to the other because the laws don't change. And I'm not trying to poke fun at this way of thinking, but I want to point that it has gone to a level, I believe, where it makes it hard to look at a truly complicated problem and say it's diverse, as opposed to saying it's complex. And so, when I took biology, it was a while ago, you learned that you had animals and plants, and then you had animals with backbones, and then you had animals with backbones with hair, and you kept on classifying. And if you look at taking this to an extreme limit, this is the case for brain cancer. This is various glioblastomas that were sequenced using the Cancer Genome Atlas. You just keep on classifying. And it's this knowledge of how these things are classified and what this implies. That's the local knowledge. And so this this clustering of this type of data that can then be manifested in various network models is more or less how people are thinking about these diseases and their heterogeneity. And one thinks if I keep on clustering, I will find different subsets of cells that maybe I can treat with different therapeutic therapies that are out there, and I can figure out the right combinations to cure a cancer. And I don't think that's the right way to think about it. And so this is another set of data. This is before it's clustered. And here you can see it's clustered in the same way, where you have organization along the diagonal. And then you can, out of this data, you can put together these similar network models. This is actually Twitter data. So it is actually just social networking data. And yet you know that, that the individuals out of which this relatively complicated looking set of patterns arise constitute a stable society. And if you look at the history of statistical physics, it's oftentimes you will have a stable system, but the individual components in that system exhibit very large-scale fluctuations. And so one of the goals and one of the successful problems that statistical physics has often tackled is how do you build that stable state from those high amplitude fluctuations. And so the concepts I want to bring here are, first, in a biological system such as a glioblastoma brain tumor, what are those fluctuations from which you can build a stable state? And what do you mean when you say you have a stable state? Okay? And I'm going to argue, and I'm going to actually show you that it works, that if you want to capture the fluctuations in a tumor, you ask, what does the tumor do at the molecular level? And the tumor basically has signals that go through that cause the cells to divide, to be insensitive to growth factor signaling, what have you. The various hallmarks of cancer, those signals are coming out of proteins. Proteins are the machines that are executing those functions. And the individual cell is the unit in the tumor that exhibits those fluctuations from which one can build out the stable state of the tumor. And so a few years ago, we developed these technologies that allow us to now to take single cells out of a tumor, statistical numbers of single cells, and to profile 10, 20, 30 different signaling proteins, say phosphorylated proteins associated with the activity in those individual cells, and to, by doing so, capture the fluctuations. So let me just talk about one set of experiments, and then I'll get a little bit more into the physics of what this kind of thing allows you to do. So what you're looking at here is it's a microfluidic chip. One can do as many as 10,000 single-cell experiments on a chip, and each of those experiments you can do as many as 10 to 30 to 40 proteins. Okay, so this is data from it's a human tumor grown in a mouse. It's labeled GBM39. This is an aggressively growing tumor. It has a particular oncogene called EGFRV3, which makes that tumor particularly aggressive. And what you're looking at here, we measured these proteins, that are represented by these balls, from individual tumor cells. And then we clustered the single-cell data. So this is a traditional biological way of thinking about this data. We're just classifying the data and looking at different functional phenotypes. Okay? And what you find is that there's 3 different functional phenotypes that are well separated in this tumor. And if you actually look at the signaling networks that you can pull out of this data, where here a blue means that these 2 proteins basically have some sort of a correlated or activatory relationship between them. If it's red, then it's an inhibitory relationship. One finds that if you look at all the data together, it more or less agrees with the literature. But if you look at any one of these individual functional phenotypes, they don't agree with the literature. In fact, you'll see that there's inhibitions here where you might have activations in other environments. Okay? And so what this tells you is that what you learn from bulk assays is not correct, that the actual details at the cellular level are very different. It also tells you that very likely, if I looked at a different tumor or a different patient, for example, I would see a different set of network interactions. Okay? And so everybody's going to be different. And also, if I look at this tumor and analyze in this way, and I say, well, how am I going to come up with a way to inhibit or drug this tumor effectively, I personally am lost when I look at this data. It does not give you that kind of insight. You might say, well, maybe we want to go to a protein that has a lot of nodes on it, and if you try that, and I'll show you, it's not going to work. And so the approach that we took is not to look at it at the level of a traditional biology, but to look at signaling modes within the tumor. And I want to give an analogy to a piano to give you an idea of what we're talking about. So if you hit a note on a piano, then you have all of this machinery that works in concert to come and bang a hammer on the string. Then you've got the soundboard, and you've got the size of the piano. All of that leads to a single mode in the piano. And if I listen to that mode and I want to damp it, I can either block the finger hitting the string. I can block many of these individual, basically anywhere in here. I can touch the string, whatever. I can silence that note. If I try to block it in multiple spots, all I'm going to do is still silence that same note. If I've hit 2 notes, I can block this one note in multiple spots. The other one I'm still going to resolve. It's still going to make a sound. Okay? And so if one thinks about the robustness, and I do believe a tumor acts like an organ. In fact, it is robust. It actually has a stable state, and I'll go into that in a little bit on why I think that's true. Then you're not going to build up a stable state from a bunch of delicate pairwise interactions. You're going to have collective responses, just like this piano. So when we look at it in this way, I lost my clicker. Here it is. We found that in the signaling coordination in these cells, there were 5 proteins that more or less dominated that coordination. Okay? That doesn't tell us anything about the modes. It just says that there's 5 proteins that dominate that coordination. When we ping this tumor with a drug, so we're not actually doing much damage to the tumor, we're just basically perturbing it, what one finds is that 3 of these proteins behave in one direction, and these other 2 proteins behave in another direction. And the implication here is that we basically damped one of these modes, and we're just resolving the other one. And so out of this very simple picture here, we make many hypotheses about how to therapeutically treat this tumor. If you take any one of these individual proteins here and use that as a drug target and hit the tumor with that drug, you won't have an effect, because basically I can damp this note, but that mode will just take the signal. If I hit any 2 targets within a single mode, it'll be the same thing. I won't have an effect. But if I hit any one target between the independent, one target from this mode and one target from this mode, I can shut the tumor down. And so we tried that. We tested 7 of these hypotheses. And every single one was right. And in fact, even in GBM is such a nasty tumor, you actually generally don't have much effect on mice when you try to treat it. But these are either individual drugs or they're independent modes. These are dependent modes, 3 different ways to drug them. And then you stop the drug and you find that you basically just shut the tumor's activity down. You haven't killed the tumor. And so I want to emphasize that out of these single cell measurements and out of these fluctuations, we come up with a bunch of different predictions, and they all turn out to be correct in this case. And they're collective responses across the tumor, and that we don't know mechanism. We don't really care about mechanism. We understand that we have a stable state of this tumor and how to perturb that state. And so let me go into that in just a little more detail and talk about trying to define the states of a tumor and how they evolve under a physical perturbation. And in this case, I'm going to talk about the case of hypoxia. So most solid tumors are hypoxic, and that means that they are starved of oxygen. And so to give you a calibration, here we are around 20% in this room. Normal tissues are at 5%. Mild hypoxia is 3%. Most tumors are generally down 2% or even below 1% at severe hypoxia. And when you have a hypoxic tumor, its fundamental wiring and its metabolism and its behavior to therapies is changed. And what we wanted to understand is how does this signaling network rearrange itself so as to give a different therapeutic responses. And we looked at, in particular, a drug that hits a protein called mTOR, because mTOR is known to be restructured in hypoxic tumors. Okay, so let me talk about fluctuations for a moment that you get at these single-cell assays and try to give you some insight into what you get out of fluctuations. So when we do these measurements, out of every single one of our assays, we'll get this sort of list of proteins we've measured. We can convert those, and we're going to use quantitative physical models, and so we convert them into copy numbers per cell. A cell, in a thermodynamic sense, is a finite system, and that means that if I measure the same protein from 2 different genetically identical cells, I will likely get a different answer. And it's when I plot a histogram of how many copies per individual cell I see by the frequency of that observation, that's the fluctuations. Okay? And of course, because I measure multiple proteins per cell, I can look at how fluctuations with one protein correlate with fluctuations with another one, and so on and so forth. And it's basically out of looking at all the proteins and measured together, not a pairwise interaction, that we get these modes that I just talked about. Okay, so how do proteins behave when you have a protein that goes through different levels of regulation? Well, an analogy I'll make again that is reasonably good analogy, like the piano, is in Los Angeles around 2 a.m. in the morning, we don't have traffic. We pretty much have it all the rest of the time. It's probably why Christoph is no longer a Caltech professor and is up here. And so at 2 a.m. in the morning, all the cars, there's nothing regulating, they're all going like 75, 80 miles an hour. There's not many cars on the road, nothing hindering traffic. But if you go out there mid-afternoon, then you might have an accident. You've got places where the car is going at dead stop or 50 miles an hour, with many, many things regulating the traffic, and your fluctuations are very broad. And so if we look at a protein in this light, and here we just have a hypothetical protein, and let's say we turn on additional regulatory processes, and here we just did a Monte Carlo simulation. And so at baseline, you have a protein that has no processes turned on, very sharp set of fluctuations. It's not involved in a lot of processes. As we turn on one of the processes, we phosphorylate, say, maybe 10% of this reservoir of protein in a certain number of the cells. And now the average level of that phosphoprotein goes up, and so on and so forth. It just statistically moves up to higher average levels and broadens. And so looking at a protein called HIF, hypoxia-induced factor, HIF, basically, as you go from normoxia to hypoxia, it goes from a sharp feature to a very spread out feature. And in fact, if you look at the literature, that's exactly what HIF does. As you go from normoxia to hypoxia, it becomes increasingly involved in various regulatory processes. And so it's like a textbook case. And in fact, you'll see it looks, we didn't intend these two, the HIF and the simulation, to look the same, but it looks almost like a textbook case for this issue. So under hypoxic conditions, HIF is regulating lots of processes. It has broad fluctuations that spread out. Okay, now let's look at these other proteins involved in mTOR complex 1 signaling. So here's mTOR itself. And so this is the experimental data of the single cell fluctuations. So as we drop the oxygen content from 21 down to 1%, you see at 21% it's very broad. And that's because mTOR is a good drug target. That's why it's regulating a lot of processes. But at 1.5%, it sharpens up. And so do its immediate effectors that are associated with mTOR complex 1. That tells you that in this little window of oxygen concentration, mTOR suddenly isn't regulating anything. And it implies that mTOR is a good drug target, except for right here. Right here, because it's not regulating anything, if you try to drug it, it's not going to do anything. And so we measured that in a mouse model. We actually had to take the tumor out and grow it as a neurosphere to get it, but it's a legitimate tumor model. And what you're looking at here are how the levels of these proteins are altered by the presence of the drug, the inhibitor. And in this little window between 1.5 and 2% oxygen, right where these fluctuations sharpen up, the drug has no effect. Okay? It turns out the same thing is true in the cell lines. And so this is a pretty surprising prediction to make. You can't drug it in this little window, but you can below and above it. And then to go and actually do it in a tumor model and see it, so why does that work? Well, it works because when you measure these protein concentrations in these cells, and you know how these proteins are interacting with the other proteins, you've begun to define how perturbations alter their chemical potentials. Okay, I know that doesn't mean much to you, but if you want to know the state of a system in a thermodynamic sense, you need to understand a certain number of the intensive parameters of that system. And if you do have that, then you can define the state of the system. And you ask, well, is that state stable? Well, if it's stable, then if you perturb it, it will respond so as to restore that stability. It doesn't have to be an equilibrium system. It just has to be stable. So you imagine this big community of Los Angeles I showed you in that satellite photo. There are many ways you can perturb it, but the city, of course, will respond. Okay? And the same thing true in these tumors. And so, in fact, you can use this relationship that puts the average copy number per cell times the protein-protein covariance matrix that you pull out of these single-cell measurements, and you can extract out this change in chemical potential. But this allows you to predict how these cells will respond to perturbations. So let's look at this in a little detail at these. So if we can predict how they'll respond to these perturbations, that means I can change the oxygen content, and I can make very specific predictions about how these protein levels will change. So here I change the oxygen content from 21 to 3%. The theory, the calculations are the red dots, and the experiments are the blue diamonds. And so you can see that if I know this data at 20%, I do a pretty good job of predicting how those protein levels will change when I go to 3%. That tells me that I have a stable state, that tumor is in a stable state, and that I understand it, and I haven't changed the state, I've just done a weak perturbation. As I change it from 3 to 2%, my prediction is not as accurate, but I still get the sign of the changes right. And that says that it's a stronger perturbation, but it's still the same state. When I go from 2 to 1.5%, this region where I can't drug the protein, you find that no longer do I even qualitatively or quantitatively agree with the prediction. That is a strong perturbation. We have perturbed the state in a way that it no longer can respond so as to restore that state. But when I go from 1.5 to 1%, I get good agreement again. So this argues that I have one state of the tumor here and one state here. And this equation tells me that it's likely that at this state transition, that the chemical potential of the proteins and their copy number are decoupled. Okay? So once again, I'm sure that probably doesn't mean a lot to many of you, but let me give you another analogy of two similar parameters. So if I have ice water is at a particular temperature, and it contains a certain amount of heat in that glass of ice water. So I have two parameters, temperature and heat. And those are corresponding parameters just like chemical potential and numbers of molecules. At ice water, if I add more heat, the temperature won't change. And it won't change because I'm at a phase transition. If I have just water, I can add more heat, or just ice, I can add more heat. And so the implication here is that this corresponds to a phase transition. And if it's a phase transition, then you can, Willard Gibbs of Gibbs Free Energy said over 100 years ago that you lose a degree of freedom. And once again, there's an analogy you can make to real life that's exact. If I boil water here in Seattle, or I boil water up in Mount Olympus at the top, Olympiad, what's it called? Mount Hood. I know Mount Hood. Mount Rainier. Yeah. Okay, Mount Rainier. It will boil at a different temperature. And it'll be cooler up high because I don't have independent control over pressure and temperature. Okay? And so we can look inside these cells and see if we lose a degree of freedom. And these are the modes that I referred to before, the modes that we identified as independent signaling modes when we drugged these types of tumors. And in fact, what you find is that the coordination mode associated with mTOR complex 1, there's actually a few of them, they all disappear in this regime. So that tells you that not only is mTOR a poor drug target in this tumor, in this hypoxic window, it's actually intrinsically impossible to drug. It's like trying to add heat and expect the temperature of ice water to go up. You can't drug it. You can't change the chemical potential and expect the number of molecules to change, or vice versa. So I'm going to just stop there and just summarize a little bit. And my point here is that, so is that there are different ways to think about complicated biological problems that don't involve looking in detailed mechanisms, but involve instead identifying states. And it's tough to do. One has to find a way to capture the fluctuations of the system such that one can define the states. It turns out these single-cell functional proteomics appear to provide a pretty good way to do that for cancer. I don't know how good they're going to be for something like understanding the overall function of the brain or how one even apply this. But certainly for understanding and predicting a little bit about how to deal with the tumor, they can be quite effective. And what that tells you is that this world of physical science that does have laws, that there are direct conduits from that into doing predictions in biology. And at least those conduits, from my understanding, are likely to be the fluctuations. And this is an example of how to do that. But there's much precedence throughout the literature, from economics to physics, in doing that. You can use those kind of measurements to make predictive, often very surprising predictions that can be experimentally validated. And I showed you that, in fact, those predictions actually do pan out. And the applications to fundamental oncology, clinical practice, what have you, I think have tremendous potential, but also into understanding what we otherwise think of as complex systems. So as chair of this session, I want to avoid the word complexity now. That's my goal. I want to think about diverse systems and challenging systems, but I want to avoid complexity.